In a single handful of soil, there's as many microbes as there are stars in the Milky Way. Today, we're going to visit my older brother at Lawrence Berkeley National Lab to learn more about soil and how they're studying it. And trust me, soil is not simple.
You can imagine it like a little city. And so there's this community of organisms that are trading different things and performing different services for each other. And the currency is these teeny tiny little molecules that are actually nanometer scale. And so trying to understand this process and how these microbes interact, how they interact with the chemistry in the soils, and how that helps benefit the plant is extremely challenging, and it requires a community of scientists to study it.
Why is it that, this is what feeds us. Why is it that we don't better understand what's going on?
Well, it's been said that soils are the most complicated living system on Earth. And so it's been very challenging to get into. And, frankly, they haven't been very sexy. But humans absolutely depend on soils.
Oh, EcoFAB, this is the heart of it.
Yeah.
What is EcoFAB?
So these are laboratory ecosystems that we're trying to build. Take what we see in the natural environment and bring them into the laboratory in a format to where they can be made reproducible and disseminated between labs. So we're using 3D printing to make the EcoFABs. So this is a mold, 3D printed mold. We fill it up with a monomer solution, cure it, and then it goes into this plasma cleaner here. Once cured, we can take it out and we put it on a microscope slide. All right, so we got the EcoFAB with a plant on it, growing hydroponically, so there's no soil in there. And it's on this microscope. And we can actually see the main root here, and there's all these fine little root hairs coming off of it. And just to give you an idea of how small these microbes are, so you can see there are these teeny tiny little blips.
This is all about how plants and the microbiome in the soil are interacting.
Yeah.
So that you can better have a more functional microbiome or a more functional plant.
Exactly. So for example, this system here, we have control over the soil, exactly what the soil is. We could even use synthetic soils, for example. We control everything about it. We can control the microbes that are in there. We can actually control the genes that are in the microbes. We can control the plant, not only the species of plants, but the genes in the plants. And we can monitor using the imaging, the sequencing, the mass spectrometry, and other technologies to see what's happening. This instrument over here is an imaging mass spectrometer. So it actually allows us to measure metabolites spatially. When we take something out of the EcoFAB and we need to extract DNA or RNA for sequencing, or we need to do some special preparation for the metabolite analysis, this robot will do it automatically.
So you take all the soup out of your EcoFAB, you put it and you process it with the robot over there, and then it goes in here and it tells you what the DNA is?
It actually sequences the super long molecule of DNA. We can determine the full sequence of an organism, like a microorganism, and use that to sort of predict what are all the capabilities that it might have. So could it make a secondary metabolite that we know signals with a plant, for example? Yeah, we've got a couple different EcoFABs here. This one has brachypodium in it.
What?
Brachypodium. You can see that we've got some soil in this. And so, this is kind of making it a little bit more complicated, where we start adding minerals and other components of soil and seeing how that affects the plant microbe interactions. Here we have switchgrass, which is one of the major biomass crops for producing biofuels. Then I also have another 3D printed object here. This is a little EcoFAB, but what's unique about this one is that we've put a mass spectrometry surface on the bottom that this NIMS chip is basically, it pulls metabolites out of the solution and kind of takes like an impression of the metabolites. And so then we can take this chip out of here after we're done with our study. And then we load it into a mass spectrometer that, in this case, it's a multi-mass spectrometer, and it uses a scanning laser beam. So it shoots the laser beam, and you can see here's the NIMS chip, and it's moving around.
Basically, you're taking the roots and everything that's happening to the roots gets imprinted onto your NIMS chip. And you take your NIMS chip out and you put it in here, and this is scanning the surface and making a chemical survey of what's there.
Exactly.
That then relates back all the way to the roots and the metabolites.
And with that, then we can make better models that are more predictive of the process, things like carbon cycling that we're interested in. We can also identify how the plants control the microbiome and how the microbiome benefits the plants, such that we could harness that to make for more sustainable agricultural practices.
You may think of soil as just dirt, carbon, and some nutrients, but soil consists of billions, actually an estimated 10 to the 30th power, of microscopic organisms communicating and working together to exchange nutrients and information with each other and with the plants growing out of it.